The overall goal of this experiment is to detect the Rab10 phosphorylation by LRRK2 using SDS-PAGE with a phosphate-binding tag.
This method can help answer key questions in the Parkinson's research field, such as how disease-linked mutations in LRRK2 affect Rab10 phosphorylation. The main advantage of this technique is that one can have a rough estimation of the stoichiometry of Rab10 phosphorylation using a simple Western blot. Many of the steps of this protocol are fairly standard and are described in sufficient detail by the text protocol accompanying this video. Here, we want to show you the steps that benefit most from visual instruction.
Prepare plates of transfected HEK2 93 cells as described in the text protocol. With the plates on ice, aspirate and discard the media. Next, carefully add 2 milliliters of DPPS to the corner of each plate and rock the plates to spread the solution. Then, remove and discard the DPBS. Next, to each plate, add 100 microliters of lysis buffer. Tilt the plate and use a scraper to spread the buffer and release the cell lysate. Then aspirate the lysates and transfer them to microfuge tubes pre-chilled on ice and wait 10 minutes. Next, collect debris-free cell lysates by centrifuging the tubes and transferring the supernatants to new tubes on ice. Then measure the protein concentrations of the cleared lysates using the Bradford assay.